Okay. So just to remind ourselves, and I'm gonna get rid of this. Whatever, I will leave it. Okay, so exam number 3, 20 multiple choice questions. I'm going to ask you to put some things in order. Like what are some things that I could ask you guys to put in order, do you think? Steps of mitosis, steps of meiosis. Anything else that we talked about that I could ask you to be like, what other processes have we talked about? All kinds of things.
Steps of cloning.
Steps of cloning, yep, about gene cloning and bacterial transformation. Could ask you about that. Who's our favorite weirdo?
Terry.
Yep, good old Terry Mullis. Why does he matter? PCR, all right. So I could ask you about some steps of the PCR process. Anything else? Any other arrange the things that you guys could think of that might be fair game? So we got mitosis, meiosis, bacterial transformation, PCR. All right. So what about mitosis and meiosis? What are some of the things that we could talk about there? I could ask you, I'm going to ask you about the similarities and differences between those two processes. How are they the same? Yeah, we got the same steps. So we got, what are our phases? Prophase, metaphase, anaphase, telophase, and cytokinesis, right? Telophase and cytokinesis, they're separate processes, but they kind of happen at the same time. So we just kind of like smoosh them together into one thing. Okay, so they do that. Why do cells do those two processes? Yeah, we got to make more cells. Okay, so let's talk about some of the differences. So specifically mitosis. What kind of reproduction do we see with mitosis? The sexy kind or the not sexy kind? For mitosis. It's not, it's mitosis is not sexy. No. So that means that meiosis is sexy, right? Okay. How many cells are produced through mitosis? 2. And what is the genetic relationship between those 2 daughter cells? They're clones. Genetically identical. Mitosis is samesies genetics. What about meiosis? How many daughter cells do we come up with? 4. What is their genetic relationship to each other? They're genetically mixed, right? That's the whole point of meiosis, is shuffling those genes. So each daughter cell is genetically distinct. What are our sources of variation, genetic variation in meiosis? What crossing over? Yep. And independent assortment. Okay, let's stick with crossing over for a second. When does crossing over occur? Prophase 1. the first thing we do. And, okay, so all right, now we know that it gives us genetic variability and we know when it happens, but what is crossing over for realsies? Like, what does it do? Why does it give us genetic variability? What's happening? Yeah, so we got our homologous chromosomes, one from dad, one from mom, and then we just do like a little sloppy doodle, right? And so maybe the alleles that we got from dad are now on the mom's, mixed up with the rest of the alleles from mom's chromosome, and vice versa. Okay. Independent assortment. I could probably ask you how that relates to Mendelian genetics. That might be like a really good essay question. Okay, so tell me about independent assortment. What does that mean? Yeah, so during anaphase, well, during metaphase, they line up all random like, right? And so they're going to be pulled to opposite ends of that pole. And so we have independent assortment twice, right? In anaphase 1, what do we have independent assortment of? That's right. So we have our homologous chromosomes that are aligning randomly, and then they're going to be yoinked to their sides or poles. In anaphase 2, now what do we get independent assortment of? The sister chromatids of each chromosome. Okay. Is there anything else we want to talk about the differences and similarities between mitosis and meiosis? I have, you know, I'm just gonna have like a little, there's gonna be a little table. You're feeling happy about that? Feeling comfortable with mitosis versus meiosis? Okay. Oh, so many possibilities for fill in the blank questions. You guys have been using that study guide that we built as a class with all the questions. So about half of the exam questions came from that study guide. So you guys can definitely look for that. If you're like, what kind of fill in the blanks questions are you guys gonna ask? Well, I probably told you about half of them. Okay. If I was gonna ask you to draw some stuff, what kinds of things would you expect me to ask you to draw? I could ask you to draw the phases of mitosis or meiosis or both. That'd be a good one. Would I ask you to draw me a Punnett square? Probably could do that.
Cell cycle.
I could ask you to draw me a cell cycle. Yep. We saw in the lecture material, we had that little clock thing. It was like most of the times interphase, and interphase is broken up into these 3 parts, and then you got mitosis. Mitosis is broken up into these 2 parts. Do you know all those parts and what's happening in all those parts of the cell cycle? Should we draw it on the board right now? Let's do it. Okay. All right. We got ourselves a little clock. That's about the middle. Okay, this big old piece of pie. Which phase should we call that? Let's call this interphase. What's happening during interphase? Yep, kind of, the cell's just doing cell stuff, right? All right. I'm gonna use a different color now. And we're gonna split interphase up into 3 parts. What should we call this part over here? G1. Let's call it G1. Why would we call it G1?
First step of cell growth, or first stage of cell growth.
The first stage of cell growth. So growth, G for growth, 1 for the first step. Ah, that makes sense. All right. So, and that's what our cell is doing there, is it's getting more bigger, right? Because it was just made, and so it has to grow up and gain some energy and do all of its cell things so that it can finally go into this phase that we're called, let's call it S. Let's call it S phase. Why does this phase make sense? It's the synthesis phase. What are we synthesizing? DNA. Yes. Yep. So this is DNA. We're replicating DNA. So what do our chromosomes look like when they're in G1 phase? They look like a Cheeto. Right? After we've synthesized our DNA, then what does it look like?
Cheetos?
2 Cheetos. And this part right here, where our 2 Cheetos are on top of each other, what should we should call that a special name? Let's get that a special name. It's kind of in the like the center, isn't it? Centromere. What is the name for this whole entire structure?
Metaphase chromosome?
It's just a chromosome, right? And so before when we just had 1 Cheeto, that was still a chromosome. But now we have a 2 Cheeto chromosome. And so we're gonna give this a name and this a name because they're special. What should we call them? Yeah, so this is a replicated chromosome, right? So a non-replicated chromosome is 1 Cheeto. A replicated chromosome is 2 Cheetos. In our replicated chromosome, each arm of our Cheeto, we're gonna call it sister chromatids. And our sister chromatids are attached to each other by the centromere. Okay, let's go back to the cell cycle. All right, we synthesized our DNA. That was a lot of work, eh? I'm tired now. So now I'm gonna have this other phase. What should we call this phase? G2, yeah, hey, look at that. Growth 1 and maybe a growth 2, right? Because now we gotta like gain all our energy back so we can do the next thing. Where's my dad? I need more colors. Okay. Now we're ready to do the next thing. Oh, and then of course our sister chromatids still look like we still got 2 Cheeto chromosomes. What's the next thing we're going to do in this cell cycle? We replicated some DNA, then we grew again. Now we're ready for something. Separation. What do we call that? Nuclear division. Right. Oh, we also give it the same. So mitosis is the same Z's as nuclear division. Okay, we're going to do that here. And then we can do all the things, right? And eventually during which of those phases?
phase.
Yeah, so we got our mitotic phase. So we're gonna go prophase, metaphase, anaphase, phase, and cytokinesis. Which of those phases do we see the separation of our two sister chromatids? The complete separation.
Telophase and cytokinesis.
So, yeah, so we're gonna say, so once anaphase starts, then they're really pulled apart, right? Because our spindle fibers are attaching to here, and they're yoinking this guy this way and that guy that way. Ugh. So it's in anaphase, we see our two Cheeto chromosomes go back to two 1 Cheeto chromosomes. And then after we've done nuclear division, which the fancy science name for that is mitosis, then we need to separate into two cells. What do we call that process? Cytokinesis. Okay, and telophase and cytokinesis are happening simultaneously, usually. Okay, so, oh, where were we at? Okay, so that was all the crap that so anything else that you guys think that I might ask you to draw? We talked about life cycles in the lecture, but that's kind of more fodder for 182. So if I ask you a life cycle question, I'll give you a lot of context, and I'll be asking a question more along the lines of, why do we see sexual reproduction? Why do we have mitosis? So rather than saying, okay, I need you to explain to me the differences between zygotic mitosis or meiosis and gametic meiosis and the other one that I can't remember right now. So I'm not going to ask you that. If you take 182 from me, then I will ask you that. Hopefully someday. Okay. Anything else that we think that we could draw?
Exponential growth.
Exponential growth. Yeah, let's talk about exponential growth, shall we? I do love me a graph, you guys. So let's say I had a graph like this that was like the number of cellular divisions. And then I had on the y-axis, let's say, the number of cells produced. Or cells in the colony. Okay, we could talk about the number of bunnies in the tortoise, right? We could talk about the number of trees, but in the context that our discussions have been so far is bacterial cells, right? So we'll keep that constant for now. But this exponential growth thing applies to any population of any organism. All right. So let's say that we have one bacterial cell. And our first, like it hasn't divided yet. So, right. So we just have one here. After it's divided one time, how many cells do we have? 2. Okay, so let's put it out right here. Okay, after a second cell division, those 2 cells are going to become 4 cells. I should probably change my axis a little bit because this is going to be. Let me make this shorter. But let's face it, the numbers. Something like that. Okay, So after one division, we're gonna have 2 cells, and then we're gonna have 4 cells, and then we're gonna have 8 cells, and then we're gonna have 16 cells, and then we're gonna have 32 cells. Then we're gonna have 64. So really, we see this. So you could draw, if I said, okay, we have a bacteria that replicates every, give me some unit of time.
20 minutes.
20 minutes. We have a cell that replicates every 20 minutes. After some other unit of time that's larger than 20 minutes, 60 minutes. How many cells are we going to have? Okay, so I can ask you that question, right? And so then you'd be like, oh shit, well, how many times would that cell replicate in 60 minutes? 3 times, right? So we would see, so we have okay, cool. And we know how many cells are produced each time it's replicated, right? How many, or like how many, what's our multiplier there? It's 2. So we're gonna have this be our base. So we could do like, okay, so we have 1 cell here, and then it's gonna grow into 2 cells, and then it's gonna go to those cells. We could do it that way, right? We could draw it out. But if we wanted to do it faster, so that we can go get out of here sooner, what's the mathy way to do this? We're gonna put this, so 2 is gonna be our base. What should our exponent be? 3. So it'd be, right, so here's where our 2 comes in. Each time we're gonna have a replication event, we're gonna double our cells. Number of cells, that's that. And then if our bacterial cell replicates every 20 minutes, in 60 minutes, it's gonna replicate 3 times. That's our exponent. So 2 times 2 times 2 is 8. Okay. So you can do it, you can draw it out, or you can do the math, whichever way works for you. Okay, and I've got big whiteboards and I've got little whiteboards, and so if you need a scratch paper during the exam, don't be afraid to ask me. Like, that's my job is to provide you the tools for your success.
What about calculators?
Sure. Yeah. I don't think we'll need them for this, but you know, if you're like just want to not have. I remember my second semester grad school. I took this class and I took the exam and I got the question wrong because I failed at basic math. So if you're like, I don't trust my ability to do math under pressure, sure. We can do calculators. Just let me know. If you're like, I'm pulling out the phone, so I can do the calculator thing. It's fine. Okay. All right, let's go back. Flip this back around. Okay. If Carson can see this, but there we go. Okay, so anything else that we was thinking that we could draw? So I could ask you to draw out the steps of gene cloning and bacterial transformation. Should we do that? Okay, let's do that. So let's just remember prokaryote. Let's just talk about prokaryotes for a minute. What do we know about prokaryotes? Simpler than eukaryotes. Because they lack some stuff, right? What do they not have, Cody? Or what were you gonna say? I guess I should ask you that question.
They can only reproduce their cloning.
They can only reproduce their cloning. And we give that cloning process a special name. We don't just call it regular old cloning,
Horizontal gene transfer.
not horizontal gene transfer. That's how a horizontal gene transfer is how we get genetic variability in these asexually reproducing things. So in order to make more cells, binary fission. Prokaryotes make more cells through binary fission, which is cloning. When you only do asexual reproduction, that usually doesn't work out well for you in the long term. So we need to have some kind of genetic variability. Prokaryotes can't do sex, so they do horizontal gene transfer. Okay, let's talk about, okay, so how many chromosomes do prokaryotes have? Not two, just one.
Circular.
Just one circular chromosome. Remember, this is going way back to like the first or second day of class, right? So the hair tie, yes, the hair tie. And so, and they bunch up that chromosome, that hair tie, into a little nucleoid region, right? It's not a real nucleus, it's just a pretending nucleus. And they also have other things, other bits of DNA that aren't really chromosomes because they're too little, and they're not, don't have a complete genome like a chromosome does for prokaryote. What's that little tiny, like little roundy circular piece of DNA called? That just floats around in the cell juice of the prokaryote. A plasmid, right? Hey, guess what we can do, because we know how to do biotechnology things, is we can create plasmids that have certain features that we want. So we're going to take our plasmid. It's a circle. We're going to make sure it has three things. What three things should our plasmid have in order to do bacterial transformation? That's not how you spell that word. Is it? Okay, cool. Okay, so we need to have an origin of replication. What else do we need to have? A restriction site. Oh my God, what is a restriction site again? Okay, so it's also called a multiple cloning site. But why do we need one in our plasmid? Well, if we have a restriction enzyme, for example, which is a special protein that goes a certain sequence of DNA, we need to have, so this is our DNA sequence where the enzyme cuts. Okay, and then the last thing is,
Resistant gene.
yeah, so we need to have a antibacterial resistance gene. Why? If you're an organism, say like an E. coli bacterium, and somebody's sticking a bunch of random stuff in you, do you want that there? But if it's beneficial, if it gives you some advantage, then you're going to hang on to that because that's a good thing. We're not going to reject that because that helps us. Okay, so here's our plasmid. We're going to take here's our restriction enzyme. Whatever, I don't care about that anymore. And we're going to snip it. And we're going to have some sticky ends, right? Now, I should make that red to be consistent. So now we have instead of having a circular bit of DNA, because this is double stranded, now we have it in a line, right? So we have a linear piece of DNA. Now what should we do? Well, the whole point of doing this is to make some protein that we're interested in, whether that's Taq polymerase, because that helps us do PCR, whether that's insulin, because that helps us treat folks with diabetes, it's cheaper. Whether that's, whatever the protein is that you think is cool, we got to put it in there. We got to put that DNA sequence, that gene in there. So we're going to introduce, here's our foreign gene. Oh. French hates the dyslexic people. All right, so we're going to introduce our gene of interest, the thing that we want to make, the codes for the protein that we want. And then, then it's just going to become a circle again. And so we're going to have our plasmid hooked up to our foreign bit of DNA, something like that. Are we done? Not quite. Because we got to take this. Oh, what should we call this?
Chimeric molecule.
This is so this is a chimeric molecule. So because it's got some plasmid and it's combined with this other thing. So it's like two things being smooshed together like a chimera from ancient lore. But it's we recombined it. Maybe we should call it recombinant DNA. Okay. So we're not done. We gotta stick that DNA somewhere in some cellular machinery that can make the protein that we want, right? Oh, look at this little E. coli bacterium right there. Let's stick it in there. Boop. This process, so now it's got its genome, right? Here's its circular chromosome that's all bunched up in its nucleoid region. And now it's going to have our recombinant DNA in there. What did we just do to this little bacterium? It's not just any normal E. coli now. It's transformed. Okay, so this process of shoving this into there is called transformation. Now we're done. Now we just sit back, grow these things, let them reproduce on their own, let them make protein, and then we harvest the protein according to whatever it is that we want. This is how we're going to make petri dish meat. Now we don't have to grow whole cows, we can just grow E. coli. Take fewer resources. Okay. Are we good with that? What else could we draw? I covered it all. Okay, I could throw you up some diagrams. Let's see. I gotta see if I can pull up some diagrams.
Like a labeled diagram.
Yeah, so if I wanted to, let me go back and pull up all of my PowerPoints. We're still in screen. let's see, gene regulation, we started with that. Hmm. Let's see. So here's what I won't do to you guys. I'm not gonna be like, what's camp? Thank you. You're welcome. Yeah, yep. And we went over this real fast one time. So the questions that I'm gonna ask you about this stuff is really conceptual. Like maybe like, when would you want to have a gene repressed? On what environmental conditions would you might not want to make lactase? Well, if you don't have lactose floating around in your cell, why would you make it a protein that breaks it down? That would be dumb, right? So it's going to be conceptual things like that. So don't worry about these details. Yeah, because we get this, we cover this real quick. So for gene regulation, though, I could maybe say, why do we see a histone shift? And then you guys have to remember what histones are. Do we know what histones are? Kind of. I'm gonna put person over here. Do we remember what?
What the DNA wraps
Yeah, it's the proteins that the DNA wrap around.
action.
Yeah, during DNA compaction. Hey, in our cell cycle, where do we see DNA compaction? Oh my God, we could put all these pieces together. At the beginning. At the beginning, right? Yeah. Yeah, you gotta pack up your China before you move out of your house. Okay. So why do we see it shift? So we don't want our DNA to get tangled, so we wrap it up, pack it up tight. But we need our DNA to make genes, to make proteins, right? So we have to do all the whole central dogma of biology. We gotta do transcription, we gotta do translation. So what's the benefit of seeing these things shift?
Access to the DNA.
Yeah, because how is RNA polymerase gonna hook on to that? It can't. So we gotta like stretch it out, and if you move over there, you move over there. We're gonna have our DNA accessible. We're gonna make our RNA. RNA is gonna go do its RNA thing, and then we're gonna end up with protein. Okay, so I could ask you something. This would be a good diagram for me to ask you about. Okay, let's see. I'm not gonna make you know about all this. I could ask you something about this, especially when we're talking about helotropy, where we have more than one gene producing multiple phenotypic effects through gene regulation. So, could I say to you, okay, so I can say, okay, write me out all the possibilities of all these exons and introns, and then you have to go, oh shit, what is an intron? Yeah, the intron is the part that we don't care about, right? This part gets slipped, this introns get snipped out before they're transcribed, but not translated. And we can remember what those things mean. And we can also splice out some of our exons. So in this case, we spliced out exon 4, and in this case, we spliced out exon 2. So I could ask you, I could give you maybe something like this, and I could say which of these sequences as possible. Because can we go 1, 2, 4, 3?
No.
No. Because of the structure of DNA from 5' to 3'. It can't go, you can't reverse it. Okay. So that's some stuff I can ask you about. Let's shoot, what else? What other kind of diagrams? I won't give you one of these diagrams. Not this time. And I won't give you that either. Okay, let's look at biotechnology chapter. Let's see what we can do here. What's fodder for here? Oh yeah, so we could talk about bacterial transformation. We drew that out on the board already. I could give you this and say and maybe cover up these steps and say what's happening here. I could do that. I could give you, oh, so here's another exponential growth possibility thing. I could say, okay, after 7 cycles of PCR, how many copies of our gene of interest do we have? It's the same concept, right? We have a doubling during each cycle. And so we can say, okay, well, it's going to be 2. In this case, it'd be 2 to the 7th power. And then you could get out your math machine and figure that out.
And that's polymerase chain reaction.
That's polymerase chain reaction. Yep.
And it's just making copies
Just making copies.
of
Yeah.
the gene. Okay.
Yes. Okay. So let's see what other diagrams might be. Oh, hey, look, I might say, hey, remember when it killed people? And I might say, okay, we know that what charge does DNA have? Positive, negative, neutral? Negative, right? So if DNA has a negative charge and we mix it with some stuff that's going to make it heavy and sink into our little wells in our electrophoresis gel, which electrode is it going to go to? It's going to go to the positive one, right? Because like charges attract each other. In gel electrophoresis, what are some of the features of a molecule that might determine how quickly or slowly that molecule will travel through our science jelly?
Size.
Size. Weight, yeah, so molecular weight and size are kind of the same thing.
Shape. Too.
Shape. Yep. So if you're a big long thing, how are you gonna go fast or slow? You're gonna slow because you're gonna be bumping into shit, right? If you're a little little small round thing, zoom, you could zip through. So I could say to you, okay, you've got a large rod-shaped molecule. You've got a large round molecule and all the different combinations, and I could say, draw them out on, so that I could go here, shoot, draw them out onto your gel. Who's going to go fastest? Okay. Those are some tricks. Here's a diagram that I won't ask you about. Okay. Let's see what else we got. I could ask you, like here's another PCR thing, right? I could say, what's happening? I could ask you this exact question. What's happening in each of these steps? And can you put them in order? Can you tell me what's happening? So we talked about exponential growth, that fodder. We talked about how I'm not going to ask you about life cycles. What's this thing called? A karyotype. What kind of information is contained within that karyotype? Or what the hell is a karyotype?
Picture representing chromosomes.
Yeah, it's a picture of your chromosomes. So can we tell from this karyotype the biological sex of this individual? Assuming this is a human. Yes, sir. Yes. What's the biological sex of that individual? Well, so it has, ooh, it's kind of, it's hard to see from the back. So here's our sex chromosome. Let me bigify it and then maybe it'll be easier. Maybe. This here says X. Is there a Y here? No. So what's the biological sex of that individual?
Male. Female.
Female. If that's a bird, then we did it with
XY
female.
is male.
What's that?
XY is male.
XY is male.
Are you gonna ask us like the sex of?
I could.
Okay.
I could say, here's a karyotype of a thing. These have X's and Y's. Chicks have 2 X's. Well, biologically speaking, not gender identity speaking. Let's say that there was another chromosome here on chromosome number 21. There was 3 dots here. And I could say, is there, there's a chromosomal abnormality. Can you identify where it is? And then you'd be like, oh yeah, there's a chromosome right there on chromosome number 21. And then if you're like a super party counts, and you could be like, that's called Down syndrome. Or that's called trisomy 21. Right? So I don't expect, what I expect is for you, if I gave you one of those and I said, there's a chromosomal abnormality, can you find it? That you would be able to find it. If you're fancy, then you know all the Down syndrome and all that other stuff. Okay, so that is a diagram that's possible. Okay, this is a diagram that's possible. We already drew this out, right? We're pros at this by now. You guys could draw me, or at least identify, because we did this in lab. And so I could say, I could take these words away and I could be like, which one of these cells is an anaphase? And then could you guys pick that out? We did that in lab, so I would expect you to be able to do that. Yeah.
I know you said like you don't really see the difference between prophase and later prophase. Are you gonna test us on the difference between those two?
No.
Okay.
So for me, there's 4 phases.
Yeah.
Or prophase, metaphase, anaphase, telophase. So these kind of transitional ones, yeah. I'm not gonna do that. But if I gave you, oh hey, look, this is what we looked at in lab. And if I gave you a picture of this and I could say, okay, where's anaphase? Could you guys find it? Yeah, I'll probably ask you an easier one like metaphase. Because metaphase is easier. So how many metaphases can you guys find? 2. I can see 2. Can you find, can you see a telophase?
Yes.
Yeah, so. Here's a nice telophase. Maybe you would call that anaphase, you know? So we would have to use our discrepancy a little bit there. But metaphase is the easiest one to pick out. Okay. Let's go back to where I was. Okay, so let's see, what else? What else do I want to ask you about? Oh, I could ask you the differences between cytokinesis and plants and animals. That would be a fair one to do. could ask you about binary fission. I could ask you about ways that eukaryotes do asexual reproduction. Let's see, let's go back to here. That's right here. Okay, so I could say, I could take away this part and I'd be like, what are the ratios of these particular genotypes? Or I could give you this and then say, okay, what are the ratios of the phenotypes? Okay, and then you would have to know the difference between genotype and phenotype.
Can you say that again?
Um,
The difference between genotype and phenotype.
Yeah, so how about somebody else wants to tell us the difference between genotype and phenotype?
Phenotype is the physical characteristic.
The which one is?
Physical phenotype.
The phenotype is physical, yes. The phenotype physical, right? These are our measurable characteristics. These are our physical traits. This is the stuff that you can see and measure. The genotype is the type of genes that you have. What are the alleles? What is the actual DNA that makes up your genome? Okay, so if I gave you this, I could say, okay, I want to know I could say, okay, so we have a cross of a dihybrid cross of parents that are both heterozygous and two traits. And I can say, what is the probability that we're going to end up with an offspring that has the genotype big R, little r, big Y, little y? Could you guys pull that information out of that chart?
Yeah.
How would we do that? So we're looking for enough more things that are heterozygous in both traits. So that, not that guy, not that guy, not that guy, that guy. Oh, yeah. Okay, let's mark him down. Put Kelly marks here. Yes. No, no, yes. No, no, yes. No, no, yes. No, no, no. Okay, and so then we can say, okay, all right, all right, all right. So 4 of those out of the 16. Oh, and then that's I could reduce that fraction down, can I? There's going to be a 1 in 4 chance that the offspring of those two parents are going to be heterozygous in both traits. Does that make sense to you, Erin?
Because heterozygous is big R, little r.
Yes. So heterozygous in this trait, heterozygous in this trait. Then we just count those bitches up. Divide by our total. So we can get genotype and phenotype from something like this. Okay. Let's see, any other diagrams of interest? That's probably good. All right, let's talk about Lindsay's favorite thing to talk about, essay questions.
Just one extra point.
Yeah, you know what? That is actually really a good point. That like, if you're even just like not super confident, but you know, write something for those essay questions, because it's so easy to get extra credit points there. And the people that don't do great, leave those at blank. I mean, the 2 essay questions this time, that's 10% of your grade for this exam. Those are so important that you write something. even if you're like, it's kind of bullshit. Welcome to college, you guys.
Bullshit.
All right, so what do you guys think would be good essay questions?
Why sex matters.
Why is sex so great, right? As I said while I was being, observed. That was hilarious. All right, so why does sex matter? Why do we see sexual reproduction? It's costly. It takes more. You're gambling, right? Could you guys all sit down and write me a paragraph about why sexual reproduction is beneficial?
All I can think about is gene diversity.
Yeah, there you go.
That's it.
Yep. Write me anything about that. And think about environmental conditions, and your environment might change. Right? We see that, we're seeing that now. And so we need genetic variability to have differences in the traits. So natural selection can be like, yeah, your set of genes is going to be good for this environment. Okay, so we also see a lot of eukaryotic organisms able to asexually reproduce. So why is asexual reproduction? Under what conditions would asexual reproduction be beneficial? So yeah, so we need to have, we need to replace our cells, right? So we need to have mitosis to be able, so that we don't just like kind of wither away. But so like when we want to make another individual, humans can't do this, but if we want to make another individual, I talked about the aphids that I studied. What are the conditions under, and fungi do this, plants do this, where they're just like, this set of genes works really well under these environmental conditions. And so I'm not going to take a chance of doing this gene recombination thing and not having my offspring be able to live in this environment, right? So I'm going to just asexually reproduce because this is a stable environment and I've already cracked the code and I'm just going to keep doing this. So you could write me a little paragraph about that. What other kind of like, so essay questions tend to be things that are like, where we're taking all different concepts and we're making connections between them. So what do you guys think would be a good essay question? And I think there was one in there that I was like, ooh, this is a great essay question.
I'm trying to find it.
Yeah. Let me just remember what essay questions I asked.
Have what is the difference between the translation and post-translational gene regulation?
Okay.
So this is a potential essay question.
Okay. So thinking about gene regulation, we have in eukaryotes, we have those different levels. And so somebody wrote the question like, what's the difference between, or like compare and contrast, translational level gene regulation and post-translational regulation. So now we have to remember what translation means. So let's go back and just remember what translation is. So we have transcription and translation. Those are our two processes. Do we remember what? So let's draw out the central dogma.
I already need a protein, right?
So central dogma is DNA, RNA, protein. Okay. So this arrow is gonna get trans something. Transcription. And then that means that this one's gonna get translation. Right, because we're going from nucleic acid language to protein language. So we're translating languages. Okay. So how can we change what proteins are being produced during translation? So we have our piece of mRNA. I showed you a picture of it before and I was like introns and exons.
RNA splicing.
We can have RNA splicing. Sure. Yep. We can have one gene and codes for multiple things. We can, so RNA is really unstable. But we can do things to stabilize RNA.
Oh, 5' cap and poly A tail flexibility.
Yeah, we can put a cap on it. We can give it a tail so that when there's environmental conditions that are breaking it down. So let's pretend that this is our RNA sequence of interest. It's going to break down our tail, poly A tail. That goes away. We don't care about that anyway. We might lose our cap, but that's fine because we care about this. And so what's that? The poly A tail and the 5' cap, what does that do to the mRNA? It's going to make it last longer in the cell, right? Yeah, in the cytoplasm. So if it lasts longer, we're going to see more proteins. But what if we don't want very many proteins? We're going to give it just a little bit of a cap, right? Or a little bit of a tail. Maybe not any at all. Okay, so those are a couple of ways that we see gene regulation during translation. What do we do after translation when now we have our protein? Right? Are we done then? We can do things like give it a little tag that says take this to the recycling. We can add extra branches to this protein. We can under different environmental conditions, that protein is going to change shape, right? These are concepts that we learned earlier in the semester, but just because we're not talking about enzyme activity specifically, those concepts still need to carry us through.
Those were the effects. Structure and function, how long the last in the cell, what the protein sound.
Yes. Yes. All of those things are good things to know. So that's a very good essay question. We talked about when sexual reproduction is beneficial and when asexual reproduction is beneficial, under what environmental conditions that we see those things happen. What are some other good essay questions? I had to come up with 4 of them. That's double as many as I had to come up with the last exams.
define budding?
So I could ask you, so defining budding might not necessarily be a great essay question, because it's like kind of trying to synthesize
Oh,
the
that's just
stuff.
short.
But yeah, it's a short answer or multiple choice answer would be, yeah, so what are the mechanisms that eukaryotes do asexual reproduction? And you could talk about budding, and you could talk about mitosis, and you could talk about fragmentation, you could talk about all this, all that stuff. So here's a pretty good one, right? Because now we're talking about inheritance, but we're also like wrapping in, bringing in meiosis. Right? So these things where we're taking things that we've kind of talked about discreetly in class, but saying, okay, how do these two things relate to each other? Just because they're in different chapters of the textbook doesn't mean that they don't jive with each other, right? So could I ask you something about about this? So something like along the lines of, in which phase of meiosis do we really see the principle of segregation? And then you have to remember, okay, well, shit, what are the phases of meiosis? And then what is the principle of segregation? And then how does that relate to inheritance? Right? So it's gonna like really push you up that pyramid to make you think more. Make you think more. Okay, and let's see the other one.
Wait, how does it relate to inheritance?
Okay, how does this relate? How does the principle of segregation relate to inheritance? We got two alleles. One comes from mom, one comes from dad, right? If we have we're gonna make a cell up here, we're gonna make a cell down here. Do these for chromosome 1, right? It's randomly decided which chromosome is gonna go where. And for chromosome 2, here's another thing that we could talk about, the multiplication rule of independence events. So chromosome 1 arrangement is independent of chromosome 2 arrangement. Chromosome 3 is independent of all those other bitches, and so forth, right? And so if we're gonna get a different, each of those 4 cells that relate or produced through meiosis are gonna have different combinations because of the principle of segregation.
And this happens during anaphase?
This happens during anaphase. Yep. So anaphase 1.
Anaphase 2?
Anaphase 2 specifically for the sister chromatids. Yep. So anaphase 1 is where we see the homologous chromosomes, our the two Cheetos, right? And then we got another 2 Cheeto guy over here. Those are going to segregate. Those are going to segregate. These are going to segregate. These are going to segregate. But in anaphase 2, then the sister chromatids are what's doing the segregation. Can you guys think of, so now if we're combined for old knowledge with new knowledge, can you guys think of any traits in humans that violate Mendel's model of inheritance? Those 3 assumptions. Well, I'm going to make you're going to have to know, if I asked you a question like that, then you would have to know those 3 things. And then you would have to think back, okay, so based on my previous experience, because I'm a grown ass adult and I've lived a long life and know some stuff, could you identify some traits in humans that Mendelian genetics is not suitable for, right? Like our traits don't follow Mendelian genetics. Can you give me an example of that? Like hair color, right? And so, okay, so then which of those three things do we think of that? Could you make an argument for that it violates those things?
Well, each gene only has 2 alleles.
Yeah, so we see a lot of variation in hair color or eye color or whatever. And so that indicates that there's multiple alleles. Or it might mean that we don't see this clear dominant recessive relationship.
Can you give an example of that? What do you mean?
Okay, so curly hair is dominant over straight hair alleles, but we see lots of people with wavy hair. So we see kind of this intermediate between, and so that means, okay, well, either there's more than one allele, or there's not a clear dominant recessive thing going on. When I first taught 181 over at NAU, I had one of my students come up to me and he was like, Julia, I'm really worried. I'm so concerned. He's like, I'm the only one in my family that has blue eyes. And I was like, well, there's possible explanations for this. And like, he was really upset that like his mom cheated on someone, on his dad. And I was like, that's possible. However, when we see this big spectrum of traits, such as eye color, there's usually more than one gene controlling that trait. And so like this kid just happened to, everybody else in his family got at least one dominant allele, and he ended up with the recessive of all the alleles. And so he's the one, he's the carrier. All right. Anything else you guys want to talk about? Any concepts here just like, good, we usually would like to go over that one more time. All right, well, I will stick around as long as anybody sticks around. So if you guys are feeling good, you're welcome to pop out. If you're wanting to stick around and ask me more questions, you can definitely do that too.
I think we already talked about this.
Let's talk about it some more.
Still not clear.
Okay.
How would bacterium regulate transcription rates?
Oh, yes. Okay, so prokaryotes. They're simple, right? They don't have a nucleus. So there's no need for there's no membrane around the DNA, right? So when they make RNA, it doesn't have to leave the nucleus because there ain't one. And so now it can just, so what we see with, okay, let's pretend that this is DNA. Okay, we've got our little transcription bubble. We're gonna make RNA, right? And then it's gonna be being pooped out. What we see is that what is that organelle that makes proteins? What's that thing called again? Ribosomes. Okay, ribosomes don't, in eukaryotes, ribosomes don't have the security clearance to get into the nucleus. But since prokaryotes don't have nuclei, they can just like be copying, they can just be producing proteins as transcription is happening. So in prokaryotes, these two processes are happening at the same time. And so if we want to regulate the proteins, the amount of proteins that are, we really just need to regulate transcription. So transcription and translation are happening simultaneously in prokaryotes. They're not separated by time or space like we see in eukaryotes.
Can you go over what the synapses does in meiosis?
So synapses in meiosis. This is really important. So do we remember what stage synapsis occurs in? Prophase 1. Okay, so the first thing that we are going to do. So we're going to have our homologous chromosomes, one from mom, one from dad. They're going to, so the way that I remember it is that the homochromos synapse together. Okay, and so what they're doing basically, instead of like being floating over here and this one's floating over there, they come and they just hang out with each other. So synapsis is just kind of the closeness and proximity of the homologous chromosomes. They synapse together during synapsis. And that allows them to do what? Crossing over.
And that also happens in prophase 1?
That also happens in prophase 1. Prophase 1 is probably one of those stages of meiosis that you're gonna really want to know, because like some cool stuff happens there that doesn't happen in mitosis. Okay, and so what does that give us? Why do we want to have synapsis of the homologous chromosomes?
More variation.
Right, this is one of our sources of genetic variation.
That's like when we took those little beads, we took the little piece of the tail off and we put it on the other one.
Yep.
Okay.
Yep, exactly. Yep, that pain in the ass.
Yeah.
You'll never forget crossing over. You still have blisters. Okay, good question. Okay, and let's just remember our other source of genetic variation. I just had it up there. And we can talk about how it relates to this principle. So our two sources of genetic variation are crossing over and
Independent assortment.
independent assortment. How does independent assortment relate to the principle of segregation?
Isn't that when inheritance happens, right? 'Cause
Yeah,
different
yeah. So
things-
it's just how, like, you know, these guys are going to get separated. It's random for each chromosome. What happens if we have two traits that are coded for on the same chromosome? Right, we didn't really talk about linked traits in this class, but if their two genes are really close together in the DNA sequence, then they're always going to either cross over with each other and segregate with each other. So sometimes we see linked traits, but we didn't talk about that, so it's not gonna be on the exam. I probably should have said that, so now because you guys are gonna be confused.
Follow-up essay question. In the
Follow-up.
presence of lactose and glucose, would we see high transcription of lactase?
Ew, okay, so,
And description.
okay, so let's do this again. Okay, in the absence, is that what you said?
In the presence of lactose and glucose, we
Okay.
see high transcription of lactate, Y, and lactate.
Okay, so we've got lactose and glucose present. Would we see, would we see the production of enzymes to break down lactose if we had both glucose and lactose present? So let's remember, so glucose, we love glucose because it allows us to do energy production real good. Okay, there we go. Lactose is a glucose attached to another sugar molecule. It's a disaccharide. Oh my God, disaccharide, remember that? Remember when we talked about that a long time ago? Right? So here's a really effed up glucose molecule attached to some other guy. If we have glucose and lactose, are we going to want to use lactose as an energy source? No, because this guy's already here. And so why would we spend more energy trying to break this bond when we've already got the thing, right? But what if we don't have glucose? This is what we want to use that, right? We would want to produce that enzyme. What if we don't have lactose or glucose? Then we're screwed. Right? So then we wouldn't make it. Okay, so that's a really good essay question where I am trying to get you guys to think critically. You know, and I wouldn't ask you about, well, the lac XYZ or whatever the hell it is, because I would just say the enzyme that breaks down lactose. And I would remind you what lactose is. Let's think about when we have our, so let's pretend we got our little DNA here. And we're going to make some RNA. There's 3 steps for this RNA polymerase. So there's 3 steps. What's the first thing that RNA polymerase has to do? So the polymerase doesn't leave the nucleus.
It's like attached.
It has to attach. What's the fancy science name for attaching? Yes.
Oh.
But in this case, yeah, in this case, it's gonna be called initiation, right? We're gonna start, we gotta start the thing. And so this is going to hook on, the RNA polymerase is gonna hook onto our DNA. Right? So then it's gonna juggle on, and it's gonna be incorporating nucleotides. And we're actually going to see the building of our mRNA. What's that process called? Elongation. Because this RNA molecule is getting longer. Okay, so now we've got our polymerase. All right, now we've reached the end. Whoop. Now we're ready to detach. What's that called? Termination. In eukaryotes, which of these three stages has the most effect on the final product of the protein that will be produced?
Initiation.
Initiation, why? Yeah, start it off. Once you start, it's gonna do it, right? But if you can prevent this polymerase from even hooking on, right? The most cost-effective way to manage a problem is to prevent it. And it's kind of the same way here, right? We might want that polymerase to be able to hook on easier if we want more proteins, or if we don't want that protein, then we would prevent that polymerase from hooking on. Right? So we either make it harder or easier for this initiation step to occur. Does that make sense? And we have promoter regions up here. The cat box and the top hat box, and the ones that are all the ones that we giggle at. That are going to actually make it easier for the RNA polymerase to hook on. Or we might have other proteins that are saying, no. There's enough glucose here. We don't need to make lactase. Right?
So would that be the repressor then?
That would be a repressor. Yep.
Was this part of the last exam or is this part of this exam?
This was in the, yeah, so this was right after we came back from spring break. This is still in gene expression where I'm saying, okay, so yeah, it did straddle it a little bit, but now in the gene expression phase, we're like, okay, how do these concepts that we learned about apply to what we do, right? Okay. So we've talked about everything during this little review sesh, that's on the exam. Yep. So if there's anything else that you guys want to talk about or need clarification for, now's the time. I'll still be here for another till 11:15.
What happens after? If I went, when the observation is what happens,
Mhm.
like what happens after?
Okay, so, it depends on what kind of critter you're talking about. So, in meiosis, we're going to come up with 4 haploid cells. Oh shit, haploid. Ooh, haploid and diploid. Oh, those are some words that we should know. Do we remember what those are? Di, what does di mean?
Double.
2. Yep. So 2 copies of each chromosome in our cells. So if we go back to that karyotype question, which is going to be really that at. Right. So here's 2 copies of each chromosome. Boop, 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 boop. So this is a diploid organism. Haploid. Yeah, 1 sec. Okay, so we've got 4 different haploid cells that are genetically distinct. Now, depending on if you are a plant, a fungus, or an animal, those cells are going to do different things. Let's say, for example, that you are a human female. Well, what will happen is 3 of those will go away. They'll just fall apart. You'll take those organic molecules and then build something else out of them. If you're a human male, each of those are going to get a little flagella, and now you have 4 little spermies to reach mitotic division. Meiotic division. Shit. Meiotic division. Okay. If you are a fungus, each of these cells is going to grow into a new individual. If you're a plant, these are going to undergo mitosis to create a gametophyte. Again, all that stuff is for 182.
So a diploid means that the cell has 2 chromosomes or
2 copies of each
2
chromosome.
copies of each
Yes.
chromosome.
So in humans, we have 23 chromosomes. And we have 2 copies of each of those chromosomes. So really, we have 46. But when we make our gametes, they only have 23 because we have it. If you are a Arabidopsis, which is a plant that we use for as a model organism in genetic studies, then they have 4 chromosomes. 2 copies of each chromosome, so a total of 8. Remember, so these are homologous chromosomes, so each location on that is going to code for the same trait, hair color, height, weight, blah, 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 blah. Ferns have thousands of chromosomes, and they have, they can be, we call it polyploidy because we're not going to count that high, right? So they can have multiple copies of each chromosome. More than 2, more than 3, more than 6. Strawberries are, I think, are octoploid, where if you looked at their karyotype, there would be 8 chromosomes for number 1. So we with our human bias tend to think, oh, 46 chromosomes, that's what everything has. No, that's just what we have. My conglets have 19 chromosomes and they're diploid.
Can you describe the difference in cytokinesis in animals and plant cells?
Sure, I can. I'm gonna go get some cell models. So let's think back to, oh God, how long ago was that when we talked about cell types? That was like months ago. What do plant cells have that animal cells don't?
Cell walls.
Cell walls, right? So if we're going to do cytokinesis in a plant cell, we got to make more cell walls.
Mhm.
That's our cell plate. And then eventually it will grow up into be a whole ding dang cell wall. But if we're an animal, all we got to do is just pinch those two cells off. And using an actin ring. Okay, actin is just a flexible protein that's going to pinch those plasma membranes off into two. This one looks like a butt to me. And so that's the cleavage furrow. And then eventually we'll have two cells that are. So plant cells, cell plate, animal cells, cleavage furrow.
Awesome.
So when they become brain cells, does that mean that you get more than variation from the male side of the family there, or?
There's a higher probability, I guess, that, well, okay, so also the thing about dudes is that you guys waste gametes like a lot. So I don't know if there's more genetic variability from the male side. That's a good question. Yeah, I'm gonna think about that a little bit. Because if we, each gamete that's produced is, like it's an independent event, just like rolling a dice, right? So, yeah, I don't know. Yeah. I think just because I think men or like males make more gametes because they can reproduce, like once we get knocked up, like that's it. Like this, we got 9 months at least before we're ready to reproduce. But like you need to knock somebody up, yeah, you can go do that 4 more times today, right? So so I think that's why we see the differences in the number of gametes, but I'm speculating. Anything else? If we're gonna use pipettes, remember those little things? We have to like put this little thing to get the volume set to the right level. But tell me about the first stop and the second stop. What does what does that even mean? And what happens at each of those stages? So if I'm gonna go, okay, I need to suck up some of this whiteboard cleaner. Got my pipette here. I'm gonna be like, oh, don't, hands, don't shake. Okay. Now I'm gonna do, what am I gonna do? Gonna go to the first stop. And I'm gonna stick my pipette thing in the thing. And then it's gonna suck up the stuff. Now I'm gonna put it in and just drink.
Oh, yum,
Now what do I do?
all of it.
I'll go down to the second stop. Okay, now I gotta get rid of this pipette tip. Eject, right? The eject button. And goes, pew, goes flying across the room.
You had it as a extra credit for question one, but what types of cells are in the bucket?
Yes, what types of cells do not undergo mitosis? Well, when I talked about them, I did this. So some nerve cells don't undergo mitosis, and some cardiac cells don't do mitosis. And so once they're made, when you're a little embryo, that they're with you till you die.
Nerve cells and cardiac cells.
Cells, yeah.
So not just the brain, but like all the nerve cells.
Well, so there's some nerve cells that do mitosis. There's some that are like really central. And I am by far not familiar with human anatomy. So
What's the other name for the nerve cells in the brain that? Or wasn't there like a name?
plants.
There was like a name though that was on the text or on the extra credit. Cardiac, and then there's like another one. Cortical.
Cortical. Yep, thank you. Nobody wanted to be the first one to leave.
So when are the extra credit papers, like the
It's,
Planet Square practice?
You can, well, send them to me as soon as you can. All right, so the extra credit is to give you practice for the exam, so let's make them do when class starts on Thursday. So the Punnett squares, the mitosis coloring thing, is that it? So if you want those little 5 extra points, it goes to me. And if you don't have them, I probably have extra copies.
Do they each have?
Yeah, each 5. You're like, I want more, put in 5 points.
I'm halfway through it. I'm like, do I really want that?
Yeah. If it's, yeah, so I
No, it's
mean,
fine.
you
I
don't
just-
have to.
My time is not.
Yeah, it is,
Like,
exactly. That's why it's extra credit.
Do you have a copy of that?
Probably.
Yeah.
Okay, I'm gonna.